In this lab, we're going to look at meiosis and human genetics. So what you're looking at is you're looking at a picture, and this large ball that you're seeing there is actually an egg cell. And all of the little cells that are attached are sperm, and the sperm are trying to fertilize the egg. And so that is how new life begins, right? We start with an egg gets fertilized by a sperm. That's going to form what we call a zygote. That one cell is going to divide to two cells to four, until we end up with a multi-celled embryo. Now, if you think about it, for egg and sperm, right, all the cells in your body that are not your reproductive cells, all the cells in your body have 46 chromosomes. And that means that you got 23 of those from mom and 23 from dad. So the cells in your body have 46 chromosomes. So when you think about reproduction and forming the next generation, right? So in me, my egg cell has to reduce chromosome number in half, meaning that I need to go from having 46 chromosomes to only having 23. And that way, if the egg has 23 and the sperm has 23, and they combine during fertilization, that's going to restore the number back to 46. And so you're going to see that the way that the cells prepare for reproduction is that they do the process of meiosis. And the goal of meiosis is to reduce chromosome number in half. So this is looking at, again, the need for meiosis. And so we talked about for lab 6, mitosis. And remember that for mitosis does PMAT, prophase, metaphase, anaphase, and telophase. And it does one round of division. The purpose of mitosis, remember, is to make two identical daughter cells. Meiosis, on the other hand, needs to reduce chromosomes by half. And so to do this, we do 2 rounds of division. The first round of division is referred to as meiosis 1. And the second round of division is referred to as meiosis 2. It's Roman numeral 2. So meiosis 1 and meiosis 2. What you're going to learn, and I'll go through the specifics in a minute, but when the cell goes through meiosis 1, we are already going to split chromosome number in half. And so notice that if you look at spermatogenesis, which is the formation of the sperm, so here is the father, right? And so in the testes, that's going to occur in the testes. So in the testes of a male, that one cell is going to undergo meiosis. And during meiosis 1, it's going to reduce chromosome number in half. So you end up with 2 daughter cells, and those 2 cells have 23 chromosomes. But after meiosis 1, they're not yet complete. And we are going to talk about why in just a minute. But each of those cells is going to undergo a second round of division, which again is meiosis 2. And so what we end up with is we end up with 4 functional sperm. And this process occurs starting at puberty. So this process begins during puberty for males, and that's when he starts to produce functional sperm. On the other end of the spectrum is what's called oogenesis. And oogenesis is the process of producing the eggs or the ovum. Now, in females, when this occurs, this process is going to happen in the ovaries. So the ovaries are the reproductive tissue. And so what's going to happen is the mother starts with a cell, and that cell has 46 chromosomes. It's going to undergo meiosis 1. It's going to split into 2 cells. Each of those cells has 23 chromosomes. But again, they're not finished yet. They're going to undergo meiosis 2. And you're going to end up with 4 cells as a result of meiosis in females. Now, the difference being that in males, you get 4 functional gametes, meaning you get 4 functional reproductive cells. In oogenesis, on the other hand, in the females, we get 1 functional egg and 3 polar bodies. And these polar bodies are support cells. They're not functional gametes. And so in oogenesis, we don't end up with 4 functional gametes. We end up with 1 functional gamete and 3 polar bodies, which are support cells. In females, females begin oogenesis when they're still in their mother's womb. So this occurs before birth. So for females, this is going to happen very early on, before a female is actually born. So if you're female, you were actually born with all the eggs you're ever going to have. Now, interestingly, when you look at meiosis in females, I said that you're born with all the eggs you're going to have. What's interesting about it, though, is that meiosis is not complete. So what that means is that the ovary is going to store eggs that are frozen in meiosis 1. So they've already began meiosis prior to birth, but they're stored stuck in meiosis 1. Now, what is going to initiate them to complete this process? Well, for females, when you hit puberty, right, your ovaries are going to release an egg every month. And that process is called ovulation. So the process of ovulation is that one of your ovaries is going to release an egg. The egg is going to travel down the fallopian tube. And during this process, it's going to continue to do meiosis when the egg encounters a sperm. So if there is sexual intercourse during the process of the egg going down the fallopian tube, the sperm are going to swim up the uterus, up to the fallopian tube, and fertilization is going to occur. And so the sperm is going to penetrate the egg. It's going to form what's called a single-celled zygote. And then the embryo is going to, that zygote is going to make its way down to the uterus, and then it's going to implant in the uterine wall. Now, if during the process of ovulation, right, if that egg does not encounter a sperm along the process, right, if there's no fertilization, the egg is not going to implant in the uterus, and as a result, a woman is going to menstruate. She's going to bleed because the uterine wall was preparing for implantation. And when that implantation does not occur, that uterine wall is going to shed, and that's what we call menstruation, or what we would typically call a period. A woman would have her period because implantation does not occur. If the embryo implants, right, and it attaches to the uterine wall, then she's not going to have her period. And that's why one of those early symptoms or early signs of pregnancy is that you miss your period, because the embryo has attached and has implanted into the uterine wall, which is going to prevent that bleeding. And so this is just kind of showing you an overview comparing spermatogenesis, so the production of sperm, versus oogenesis, which is going to produce the eggs. Males will produce 4 functional gametes, so 4 functional sperm, and this process doesn't occur until puberty. Females, on the other hand, will form 1 functional egg, 3 polar bodies, so 3 support cells, and this occurs in females not just at puberty, but actually before she's even born. And so those are some differences between these two processes, but the overall goal is the same. And that is that we need to reduce chromosome number by half. So in order to understand about meiosis, we need a couple terms. And so the first term that we need to understand is what we call a homologous chromosome. And homologous chromosomes, we get one from mom and one from dad. And so for you, right, you have 46 chromosomes and they are 23 pairs. So 23 of your chromosomes came from mom, 23 of your chromosomes came from dad. Now, how do we know which ones are the homologous chromosomes? Well, homologous chromosomes are matched in length. So if you look at what's called a karyotype, where you can see all the different chromosomes, you'll notice that chromosomes vary in length. Some are a lot longer, some are smaller, but when you look at a homologous pair, they're matched in length. They're the same size. The other thing that you're going to notice is that they have the same centromere position. Now, what that means is that if we were to look, so here's this red one. This red one is actually going to be linked to another red one, and it's going to be linked together at what's called the centromere. And that is a sequence of DNA that proteins come in and it holds those two pieces of DNA together. Now, those two pieces of DNA that are held together at the centromere, those are called sister chromatids. And sister chromatids are exact copies. Because you might recall when we talked about mitosis, remember that in interphase has G1, S, and G2. Remember that in S phase, S is for synthesis, it's for DNA synthesis. That is going to be where DNA is duplicated. It's going to be made an exact copy. So if I look at this particular chromosome, notice that this chromosome contains the gene for eye colors. And eye color can have different versions of a gene, which we call alleles. I'll talk more about that in a minute. But let's say, for example, that a person has one brown-eyed gene and one blue-eyed gene. So notice on the maternal chromosome, meaning the one that comes from mom, the mom has the brown-eyed gene. And so at that sister chromatid, because they're exact copies, at the same position on the sister chromatid is the other gene that contributes to brown eyes. Now, if we compare that to its homologous pair, so here is the paternal chromosome, and we're going to draw its sister chromatid. And again, what makes homologous chromosomes the same is that the position of the centromere is the same. So notice that at the same position on the chromosome, you're going to see that centromere. What you'll notice if you get a chance to look at a karyotype is that sometimes centromeres are in the middle, but not always. They can be off to one end of a chromosome and not in the middle. But within a homologous pair, the centromere is in the same position. Now, notice that on the homologous pair, so if I look at the one from mom, right, has the brown-eyed gene. The one from dad has the blue-eyed gene. So on the one from dad, on the sister chromatid, right, because the sister chromatid is the exact copy, on both sister chromatids, you're going to find the gene for blue eyes because, again, they are exact copies. So homologous pairs are matched in length. So the one from mom and the one from dad are the same length. They have the same centromere position. So the centromere position is in the same place. They have the same, what we call banding pattern. And that is that when we stain those chromosomes, they have regions where they compact in tight, and you see these darker bands, and you'll see parts that are lighter bands. And so they have, homologous chromosomes have the same banding pattern. And lastly, they have the same gene location. Now, the position of the gene on the chromosome is referred to as that chromosome's locus, or plural would be loci. So if we're talking about multiple locus, we would call that loci. So if we're looking at the gene for eye color, on this chromosome, let's say this is chromosome 11, okay? If we look at this chromosome at the same position on the homologous pairs, you're going to find the gene for eye color. So it's not like you're going to find one at the top and one in the middle. On the homologous pair, those genes are going to be at the same position. They are not necessarily identical, though. They can be, what we call alleles, alternate versions of a gene. So notice that in this case, both genes code for eye color. One gives brown eyes, though, and one gives blue-eyed. Those are alleles. They both code for eye color, but they're alternate versions of a gene. One is going to cause cells to produce a brown pigment. The other is going to cause cells to produce blue pigment. And so that's the gene for eye color. Now, you need to realize that obviously this is very dramatically oversimplified, right? There are not just two eye colors. There's not just brown and blue. There are lots of variations even within brown and blue. There are colors in between. There's green, there's blue-green, there's light blue, there's dark blue, there's light brown, there's dark brown. There are a lot of other eye colors. And so what that tells us is that eye color is actually not inherited by just one gene. There are actually multiple genes that code for eye color. And depending on which combination you have is going to dictate what eye color you have. Now, if we look at these alleles, alleles of the same gene are written with the same letter. So let's say we use the letter B for the eye color gene. Brown-eyed could be big B, big B. I'll explain what that means in a minute. The blue-eyed gene is going to be recessive, which I'll talk about in a minute, which means that it's going to be little b, little b. So now let's write definitions for dominant and recessive. So we need a few terms. So if we say that an organism is homozygous, homo refers to same. So if we say homozygous, that means that they have the same alleles on homologous pairs. So like if somebody had two brown-eyed genes, we would say that they are homozygous for eye color. They have two of the same alleles. So homo, like think of homosexual, somebody who likes the same sex. So homozygous means they have the same alleles. So then what do you think heterozygous refers to? So heterozygous refers to different alleles. So in this example that we're looking at, this individual we would say is heterozygous because they have two different alleles. Now, when we talk about what genes somebody has, their genes are going to be their genotype. So genes that organism has. So in this case, this genotype is going to be big B, little b. Because one of the chromosomes has a big B, the other homologous pair has a little b. So that is their genotype. That's what genes they have. Now, phenotype, on the other hand, so phenotype is going to be the physical characteristic the physical characteristic that is seen. So meaning it's the physical consequence of having a particular genotype. So what that means is that in this case, the genotype is big B, little b. That's the genes that it has. Its phenotype is going to be what actual eye color that gives. So in this case, a brown-eyed gene and a blue-eyed gene, the phenotype is going to be brown eyes. So the genotype is what genes they have, so big B, little b. And the phenotype is going to be the physical consequence or the physical characteristic that is seen, meaning it's the outcome of what the genotype is. So if you are big B, little b, your phenotype would be brown eyes. That's what you see. Now, in terms of dominant and recessive, we need definitions for dominant and recessive. So let's put that on here. So if we say that an allele is dominant, that's the allele seen in heterozygous. So notice that the brown-eyed gene is given the big B, the capital letter. That's because brown eyes is dominant over blue eyes. If you have one brown-eyed gene and one blue-eyed gene, right, one brown and one blue, the eye color appears brown. So brown is the dominant allele. On the flip side, the recessive allele is the allele not seen in heterozygous. So the allele that's not seen in the heterozygous. So in this case, blue eyes is the recessive allele. It's there, but you don't see it, right? The phenotype is not blue eyes. The phenotype is brown eyes because brown is dominant to blue. And this is why if you ever think about if you know people who have two parents with brown eyes, is it possible for two parents with brown eyes to have a blue-eyed kid? And if you've ever seen this, which actually does happen, right? The answer is yes, that does happen. Because two parents who have the dominant allele could carry the recessive allele, meaning that they're heterozygous, you don't see the blue-eyed allele, but they still have it. And so if those two parents both carry the blue-eyed gene, and just by chance they both give the blue-eyed gene to their offspring, even though both parents have brown eyes, the offspring would have blue eyes. So that's why you can have two parents that don't have a trait, and then you see that trait appear in the offspring. It's because the parents are carriers for the recessive allele. You don't have it, right? You don't see that allele, but it's still there. And so that's the difference between dominant and recessive. And so we have terms depending on if a cell has its homologous pair or not. So if a cell is referred to as being haploid, think of haploid half. They have half their chromosome number, meaning that these are cells with one set of chromosomes. And the way that you can identify if a cell is haploid is that haploid cells do not have their homologous pair. So if I look at these cells on the right, you can see that we have, so let me put on my pointer here. So you can see that in this cell, this is one chromosome. This is another. Again, the black is representing the centromeres, what's holding the sister chromatids together, right? So those two pink held together, those are sister chromatids. The two blue held together, those are sister chromatids. But is the pink chromosome and the blue chromosome, are those two chromosomes a homologous pair? So think about what we said, what criteria have to be met in order for them to be a homologous pair. And that is they have to be the same size. Notice this pink one is larger than the blue one. They have different centromere positions. The blue one is off to the edge. The pink one, the centromere is in the middle. So this pink one and this blue one, these are not homologous pairs. The pink ones represent the ones that are maternal, meaning the ones from mom. The blue ones represent the paternal, which are the ones that come from dad. And so notice that this cell is haploid. It does not have its homologous pair. It has half of its chromosome number, right? Here's one chromosome, here's two. So both of these cells on the right are haploid because they don't have their homologous pairs. Now, which cells in the body are going to be haploid? Well, the answer is going to be your gametes. Your egg and your sperm are going to be your cells that are haploid. And that's because, excuse me, we need to reduce chromosome number in half. So in humans, right, in humans, what are called somatic cells are all the other cells in your body that are not gametes. So skin cells, liver cells, muscle cells, those are all types of somatic cells. And so in human somatic cells, our cells have 46 chromosomes. In our gametes, we have 23 chromosomes, right? Chromosome number is reduced in half because they do not have their homologous pair. So when we call a cell diploid, di means two. That means that it has homologous pairs. So notice that it says cells with two sets of chromosomes, meaning that the cell has the homologous pair. So if I look at the cell on the left, see if you can find where the homologous pairs are. And so I'm going to color code them for you. So this and this are a homologous pair. They're the same size, the same centromere position. Whereas this and this are a homologous pair. They're the same size, same centromere position. Notice that for each homologous pair, one comes from mom, it's a pink chromosome. One comes from dad, it's a blue chromosome. So this cell on the left is a diploid cell. Di, again, means two. It has its homologous pairs. So what cells in your body are diploid? Well, all of your somatic cells, which are basically all your cells except your gametes. So all the other cells in your body have 46 chromosomes. Now, you'll see that written also, gametes are N equals 23. So that means that there are 23 chromosomes in a haploid cell. Diploid, notice, is 2N. So that's 2, N is 23, so 2 times 23. Diploid cells have 46 chromosomes. Notice in the example on the bottom, right, the haploid cell says N equals 2. There are 2 types of chromosomes in that cell. Whereas the diploid cell says 2N equals 4 because there are 4 chromosomes in those cells. So haploid is half. So again, if you're trying to figure out if a cell is haploid or diploid, look for the homologous pair. If it does not have its homologous pair, it's haploid. If it has the homologous pair, it's diploid. So again, this is an overview of kind of why the body needs to do meiosis. And so again, we start out, here's our multi-celled diploid adult. So in your body, all the cells in your body have 46 chromosomes. However, in your reproductive cells, so females for ovaries, males, testes, in those reproductive tissues, specialized somatic cells in those tissues will undergo meiosis. So they will reduce the chromosome number in those cells from 46 down to 23. So for females, they're going to undergo meiosis and produce the egg. The egg is haploid. It has 23 chromosomes. The males are going to produce sperm in the testes. And so again, specialized cells in the testes are going to undergo meiosis, and they're going to give rise to sperm, which are haploid. Now, fertilization is where egg and sperm come together. And so egg and sperm are going to come together. And when egg and sperm come together during fertilization, you're going to end up with a diploid. Di means two, because now you have 23 from mom and 23 from dad. You have your diploid zygote, and that single-celled zygote has 46 chromosomes. So now we've returned chromosome number back to where we started. And so this is why we have to do meiosis, because if you think about it, if your cells didn't reduce chromosome number in half, and your egg cells or your partner's sperm cells both had 46, right? If they didn't reduce chromosome number in half, and then those cells came together, well, then the offspring would have 92. And then when their offspring, then they'd increase the chromosome number more. So in order to keep the number of chromosomes constant, in meiosis, in reproductive cells, we need to reduce chromosome number in half, so that when fertilization occurs and egg and sperm come back together, we get back to being a diploid zygote. Now, that one cell is going to undergo mitosis, which is what we learned last time. Mitosis, remember, is taking one cell and making exact copies. So that one cell is going to divide, and it's going to give rise to two cells. Those two cells are going to divide and give rise to 4 cells. 4 cells divide and give rise to 8 cells, and so on and so forth. And so the way that we end up being a multi-celled adult is because of mitosis. And again, this goes back to our lab on mitosis. Remember that I asked a question about, do all the cells in your body have the same DNA sequences? And the answer is yes, because all those different tissues in your body arose by mitosis. They arose because that one cell divided to 2, those 2 cells to 4, and so on and so forth. So all the cells in your body, excluding your reproductive cells, have the same DNA sequences. And that's because they arose through mitosis. What makes those cell types different, again, is going to be what we call differential gene expression. Which genes are turned on or off? So again, in skin cells, you're going to see the gene for keratin turned on. In red blood cells, you're going to see the gene for hemoglobin turned on, and so on and so forth. And so all the cells in your body have the same DNA sequences because all those different cell types arose because of mitosis. So mitosis is for, think of asexual reproduction, so without a partner. So single-celled organisms can reproduce by asexual reproduction. Mitosis is again also used for growth and development, as well as repair of tissues. Meiosis has a different purpose. Meiosis is used for sexual reproduction, and it's used to reduce chromosome number in half, so that when gametes come back together, we restore diploid number. So we're going to start by going through an overview of how meiosis works. And so again, we need some terms and what happens in each of these cells. So just like mitosis, meiosis is going to begin with interphase. So G1, S, G2. So what this is showing you, this cell here is in G1. So it's before DNA is duplicated. So what we're going to track in this case is number of chromosomes and number of DNA molecules. So I'll walk you through this. So number of chromosomes, number of DNA. All right, so this first cell that's in G1, it has 2 chromosomes. So this blue one and this red one, those are separate chromosomes. They're long linear pieces of DNA. So there's 2 chromosomes and there's 2 DNA molecules. So each chromosome at this point is made up of 1 linear DNA molecule. So we're starting out, this original cell, notice, is diploid, right? The cell is diploid because you can see it has its homologous pair. The cell is going to go through S phase and chromosomes are going to duplicate. So now we end up with our sister chromatids. So this would be like a cell in G2, for example. So notice that now they have their sister chromatids. So the 2 blues are held together at a centromere and the 2 reds are held together at a centromere. So our number of chromosomes is still 2 because when these are held together, that still counts as 1 chromosome. And so we have 1 chromosome, we have 2. But our number of DNA is now 4 because they're duplicated, right? So this is 1 chromosome, right? So each one of these is 1 chromosome, but 2 DNA. We have 2 of those, so in this cell, number of chromosomes is 2, but we have 4 DNA. So cell starts with a diploid cell. That diploid cell starts out unreplicated, so like in G1. S phase is going to happen, chromosomes are going to duplicate, they're going to end up with sister chromatids. Now, once interphase is complete, the cell is ready to go into meiosis 1. And so what's going to happen in meiosis 1 is that homologous chromosomes are going to pair up. So you might recall that when we learned about mitosis, that chromosomes line up single file, meaning they line up so that one sister chromatid faces one side and the other faces the other side. Meiosis is a little bit different. They don't line up single file. Homologous pairs are going to find each other and they're going to line up in the middle. What separates in meiosis 1 are going to be our homologous chromosomes. So meiosis 1 separates homologous chromosomes. So notice that when this cell divides into 2, the blue one goes one way, the red one goes the other way. So what we end up with is our number of chromosomes in our offspring. The cell has 1 chromosome, but those chromosomes are still duplicated, so number of DNA is 2. So, something to think about. If I look at the end of meiosis 1, right? If I look at the end of meiosis 1, are those cells haploid or diploid after meiosis 1? And so if you look at that cell after meiosis 1, does it have its homologous pair? The answer is no, which tells us that the cell is haploid. It has reduced its chromosome number in half, right? If I look back to the very beginning, my chromosome number is 2, my DNA number is 2. If I look at the end of meiosis 1, my chromosome number is reduced in half. I have 1 chromosome, but I still have 2 DNA molecules. So I have not reduced DNA number in half. So this is why we need to do meiosis 2. And so there's a question in your lab that asks about that. Why do the cells need to undergo meiosis 2? Well, the answer is cells are already haploid. So haploid cells, but sister chromatids, so sister chromatids are still together. So sister chromatids are still together, meaning they have their duplicated chromosomes, and DNA number is not half, right? So after meiosis 1, we have reduced chromosome number because our homologous pairs split, but our number of DNA is not reduced yet because we still have 2 DNA because each chromosome is still duplicated. So each chromosome has 2 DNA molecules. So this is why the cell needs to undergo meiosis 2. So we need to undergo meiosis 2 to separate sister chromatids. Now, you might recall that in mitosis, right, mitosis is going to split sister chromatids. So meiosis 2 is just like mitosis. So now the chromosomes are going to line up single file, and sister chromatids are going to separate. And so when they do, if you look at the end, we end up with 4 cells. Those 4 cells, so each cell, so if you look at one of these cells, number of chromosomes is 1. Number of DNA is 1. So notice that by the end of meiosis 2, we end up with 4 haploid cells, right? So they don't have their homologous pair. Chromosome number has been reduced in half. And we've now reduced DNA number in half. So this haploid gamete is now half of where we started. And so this is how meiosis works. Now, a couple interesting things. Between meiosis 1 and meiosis 2, the cell doesn't go through interphase again. So it's not going to go PMAT and then interphase and then PMAT again. It's just going to go directly from meiosis 1 into meiosis 2. So that's something interesting to know. The way that I can remember what separates in meiosis 1 and what separates in meiosis 2, for me, the easiest way to remember what separates when, alphabetical order. Homologous chromosomes separate first, sister chromatids separate second. So H comes before S. So homologous chromosomes separate first in meiosis 1. We end up with haploid cells, but they're still duplicated. So we have to go through meiosis 2 to split those duplicated sister chromatids. And so again, this is why meiosis 2 is going to be like mitosis, because mitosis is also going to split sister chromatids. And so this is just an overview of what happens in meiosis. So if we were to compare spermatogenesis and oogenesis and look at haploid or diploid, right? So this would be like a cell in G1, right? And in the cell in G1, notice, do we have a homologous pair? And the answer is yes. So this cell is diploid, which again, we can write 2N. So it has its homologous pair. The cell is going to undergo S phase, and it's going to duplicate its DNA. And so for this cell, which is like in G2, for example, this cell is still diploid, right? It's still diploid because it has its homologous pair. Notice we're separating homologous pairs. So this is going to be meiosis 1. Happens here and happens here. So meiosis 1 is going to separate homologous pairs. So our offspring, so these cells, are they haploid or diploid? And the answer is they're already haploid because those are not the same size. So those are not a homologous pair. So the cells are already haploid. However, they still have their sister chromatids. And so that's why we have to undergo meiosis 2 to separate sister chromatids. And so our cells are going to end up still haploid, right? because they don't have their homologous pair. And now, so this is meiosis 2. And now they also are unduplicated because the sister chromatids separated. Now again, this one cell is going to mature into a mature egg cell. So when it matures, it's still going to be haploid. If we do the same thing looking at spermatogenesis, or the production of sperm, so again, we start with a diploid cell. We have S phase, just like on the other side. This cell is still diploid. We undergo meiosis 1, homologous pairs split. Cells are haploid. Meiosis 2, sister chromatids are going to split. Sperm cells are still haploid and now unreplicated. And then all 4 of those offspring, all 4 of those gametes are going to mature into mature sperm. So that'll still be haploid. And so this is just comparing, again, oogenesis and spermatogenesis. Spermatogenesis is going to produce 4 viable gametes. Oogenesis is going to produce 1 gamete, 1 egg, and 3 polar bodies, which are support cells. So now, if you think about your cells, right? Your cells have 46 chromosomes, right? So 46 linear DNAs, which again means that they have 23 pairs, right? So on the left, we have a homologous pair. And on this particular homologous pair, we have the gene for eye color. And again, brown eyes is going to be dominant over blue. So we're going to give this big B, and we're going to give this little b. Okay, so we have big B, little b. Now, on another chromosome, right, on another chromosome, we could have the gene for hair color. And that could be red or not red. Again, that's oversimplified because just like eye color, there's a whole range of different hair colors as well. So again, this is very oversimplified, but we will go with it. So for hair, we're going to do, I don't want to do B because we did that for eye color. So I'll do H for hair. So black hair is dominant over red hair. So we'll give this a little h. So one of the things that we need to understand is when we do meiosis, right? Remember that what's going to happen in meiosis 1 is that homologous pairs are going to separate, right? One homologous pair is going to go one way, the other goes the other way. You have 23 pairs. It's not like your chromosomes talk to each other and they say all the ones from mom on one side and all the ones from dad on the other side. That doesn't happen. Each homologous pair lines up independently of any other homologous pair. And so that leads to more genetic variation, which you're going to see in a minute. That's what we call independent assortment, that each homologous pair assorts independently. It's not that all the mom ones go one way and all the dad ones go the other, right? When you have kids, you give a mixture of your chromosomes that came from mom and dad. So I might give my kids certain traits that came from my mom, or I might give certain traits that came from my dad. My kids have a mixture of my mom and dad's DNA. It's not like my kids have all the ones from my dad or all the ones from my mom. It's a mixture. And that's because each homologous pair lines up independently. And so let's look at that. So meiosis 1, in prophase of meiosis 1. In prophase of meiosis 1, homologous pairs are going to come together. And they form what are called tetrads. Tetra refers to 4. So it's 4 DNA. So notice that in this hypothetical example, by chance, you could end up with the ones from mom lining up on one side and the ones from dad lining up on the other side. And remember that this would be big B for brown, little b for blue, and black hair is dominant, so big H, little h. And so when they go through meiosis, right, homologous pairs are going to split, and then sister chromatids are going to split. And so you end up with a gamete that has the brown-eyed gene and the black hair gene. Or this gamete can have blue eyes and red hair, so little b and little h, right? So those are two different gametes that can form. Now, if you were to look at a different example, right? A different example. So if I look at this example, right? And instead of the ones from mom lining up on the same side, what if this bottom pair was reversed? So that when these separated, this gamete could have brown eyes and red hair. And this gamete would have blue eyes and black hair. So notice that because of independent assortment, because of the ways that these can line up, those will give you unique gametes. This first gamete has brown eyes and black hair. The next one has blue eyes and red hair. The next one has brown eyes and red hair. And the next one has blue eyes and black hair. And so this, again, is going to be independent assortment. Now, if you think about this, so for this one, there's two homologous pairs. So what that tells us is that there are 4 possible gametes. If you're trying to figure out the number of different gametes, you can use a formula that is 2 to the n power, where n is the number of homologous pairs. And so what that comes out to, for us, how many homologous pairs do you have? And the answer is you have 23 homologous pairs. So 2 to the 23rd, let me erase that because that went a little crazy. 2 to the 23rd power, that is the number of different types of gametes that you can produce. So that doesn't include crossing over, which we'll talk about more later. Simply by independent assortment, 2 times 2 times 2 times 2, 23 times 2 to the 23rd power is the number of different gametes that can be produced. Now, that's in my body, right? So 2 to the 23rd power. So 2 to the 23rd power, let's see if we can. So 2 to the 23rd power, if I recall properly, it's approximately 8 million different combinations. My gametes can produce 8 million different options, so 8 million different gametes. My partner, who's producing the sperm, can produce 8 million different options. So if you multiply those together, you end up with trillions of different possibilities of the ways that the chromosomes can rearrange. And so that just kind of shows you that just by independent assortment alone, you're always going to have genetic diversity. This is why you and your siblings are not identical. Yes, you may have a lot of traits in common, right? Because you're getting genes from 2 parents, but you're not identical. And you're not identical because there are trillions of different combinations that are possible. The chance that 2 people would acquire all of the same chromosomes, never going to happen. Never, ever going to happen. The only exception to that is identical twins. And that's because if you think about twins, identical twins versus fraternal twins, right? Identical twins are identical. The way that identical twins form is that early on in development, that one cell divides to 2, and those 2 cells divide to 4. Early on in development, that one embryo actually splits into 2. And when that one embryo splits into 2, that leads to identical twins. They're identical because they came from the same fertilized egg. They have exactly the same DNA sequences. That is different than how fraternal twins arise. So if you think, for example, if you think of boy-girl twins, right? Boy-girl twins. Can boy-girl twins ever be identical? And the answer is no. It's not possible, right? Because boys have XY and girls have XX. They can never be identical. They're always fraternal. Fraternal twins are not the same as identical twins. The reason that they're different is that in identical twins, remember I said that one embryo splits into two. The way that fraternal twins arise is completely different. Normally, each month, a woman ovulates and releases one egg. Now, sometimes the body releases two eggs. Maybe both ovary release an egg. If two eggs are released and both eggs get fertilized and both of those fertilized eggs implant, you end up with fraternal twins. Fraternal twins have no more in common than any other two types of siblings because they're each a unique egg and a unique sperm. So they have just as much genetic variation as any other two siblings. The only difference is they share a birthday. But the only way that you're going to end up with siblings that have exactly the same chromosomes would be identical twins. And that's because they arose from the same fertilized zygote. That's in contrast to other siblings where just by sheer chance, it's never going to happen that two offspring end up with the exact same combination of chromosomes. So now we're going to look at some human traits. But what you need to realize about this is that a lot of these traits, again, are overly simplified. And you recall that when I talked about eye color, for example, right, and we were talking about brown being dominant over blue, again, that's an oversimplification because you know there are many more eye colors than just brown and blue. So keep that in mind when you're looking at these human traits. Some of these are definitely oversimplified. So what you're looking at is comparing different types of human traits. And so what I did was I gave you some pictures to show you what that would look like. And I also put a box around the dominant trait. So if we look at hairline, for example, if we look at hairline, if the hair comes to a peak, we call that a widow's peak. If the hairline is straight across, we call that a straight hairline. If we're talking about the ability to roll one's tongue, think of it like making your tongue into a taco. If you can roll your tongue into a U or a taco shape, you have the ability to roll your tongue, and that's dominant over the inability to roll your tongue. Now, think about this for a minute. If we say that you are unable to roll your tongue, so you cannot take your tongue and put it in a taco, do you know your genotype? Notice there's no box around it, so that's the recessive phenotype. And so if you're unable to roll your tongue, that means you do know your genotype. You would have to be homozygous recessive. On the flip side of that, if you can roll your tongue, so like I can roll my tongue, I can make it into a taco. If I can roll my tongue, just by that information alone, do I know my genotype? And the answer is no. I could be homozygous dominant, or I could be heterozygous. Just by looking at me, I don't know my genotype. What other possible information could I use to determine my genotype? Who else in my family could I potentially look at? And the answer could be I could look at either my parents or my children. Because let's say I have a child, right? And my child is unable to roll their tongue. That means that my child is homozygous recessive. They have two little letters. Let's just say we're giving it the letter A. If my child is unable to roll their tongue, my child is little a. And what that tells me is that if my child is little a, then both parents had to give a little a. So if I could roll my tongue and I have a child who can't, I know I'm big A, little a. I have to be because my child got a little a from me. So for some of these traits for the dominant, you often don't know your genotype. Again, you can look at your parents, you can look at your offspring, your kids, and that may give you information about your other allele, but it might not. So you have to keep that in mind. Another trait that we're going to look at is mid-digital hair. So mid-digital hair is referring to not this knuckle, not between the first part where your finger bends, but hair on the second fragment, so the middle part of your finger. If you have hair on even one of those fingers in the mid-digital part, the middle part, if you have hair on that, you would have mid-digital hair present. That is dominant. If you do not have hair on that mid-digital region, we call that mid-digital hair absent. That's recessive. Hitchhiker's thumb. Hitchhiker's thumb is if you can bend your thumb back to, close. Mine can go back to about almost a 90-degree angle. And so if you can bend your thumb back like this, you have a hitchhiker's thumb. If you cannot, if your thumb does not bend like that, then you have a straight thumb. Straight thumb is dominant. Hitchhiker's thumb is recessive. Looking at the bridge of your nose. Some people might have a straight bridge. Some people might have a convex bridge, which means they have like a little bump on the bridge of their nose. Convex nose is going to be dominant over having a straight nose. Here's another set of traits, and the traits are looking at your earlobes. Notice there's two differences. So if your ear goes straight across, we call that attached earlobes. That's recessive. If your ear comes up to a point and the bottom of the lobe is not attached, we call that a free earlobe. So it's free, it's hanging down, it's not attached. Free earlobes are dominant over attached. Now, hair, right? So if your hair is curly, wavy, or straight, notice there's more than two phenotypes. Hair type is what we call incomplete dominance. Incomplete dominance means that the heterozygous is in between the two. Now, remember when we talked about letters, right? And we said that if we're calling a trait dominant, right? So like we said this one, so rolled tongue. Let's say we gave this an A. So this would be big A something. And inability to roll the tongue would be little a, right? If we're talking about incomplete dominance, neither trait is dominant over the other. And so as a result, giving it an uppercase and a lowercase designation does not work because that's saying dominant and recessive. Incomplete dominance doesn't work that way. So the proper way to have notation for this would be, let's say we're talking about curly. We could have the hair gene, and it has the C allele for curly, and the other hair gene, and it has curly. Straight would be HS, so the two straight alleles. And then if you inherit one curly and one straight, then your hair would be wavy, which is in between being curly and straight. This is incomplete dominance because when you're heterozygous, when you have one curly allele and one straight allele, when you have that combination, your phenotype is halfway in between. It's not curly, it's not straight, it's somewhat curly, it's in the middle, it's wavy. And so this would be an example when you see an example like this where there's three traits. What that usually tells you is that that trait displays incomplete dominance and that the heterozygous has a phenotype in between the other two. And so again, we don't give them a capital and a lowercase. We can give them the gene. So let's say we called this gene H, HCHC, which means your two curly haired alleles. Straight would be HSHS, two straight alleles, and wavy would be HCHS. Now, I cannot simply write this also. You don't want to write the nomenclature like this: CS, CC, and SS. That is wrong. You never want to do that. And the reason for that is when you see in this heterozygous, the CS, because they're written as different letters, that indicates that they're different genes. This is not. This is the same gene, but with different alleles. So that's why I gave the gene the H, and then I included the superscript, which is the allele that it has. So just know, be careful how you use your nomenclature for the incomplete dominance. Writing it with different letters is not the proper way to do that. So another example of incomplete dominance in humans is a condition known as familial hypercholesteremia. And what that means is that is genetically high cholesterol. So if high cholesterol tends to run in your family, like multiple generations have high cholesterol, that usually indicates that there's some genetic component to that, meaning that there's something that is inherited that is causing that high cholesterol. So how does this work? Well, normally your cells have a type of receptor. You can see that in the purple, which is called LDL receptors. LDL stands for low density lipoproteins. These LDL are the receptors, and their job is to bind to LDL. Now, what does LDL do? LDL is our cholesterol carrier. It's going to carry cholesterol. In the case of LDL, it carries cholesterol towards the heart. So your liver makes the cholesterol, and then it's transferred to the cells in your body because your cells take in cholesterol for several reasons. One is cholesterol serves as the building block to make steroid hormones. So your body converts cholesterol into testosterone or estrogen, etc. The other reason that your cells need cholesterol is that cells use cholesterol in their cell membrane because cholesterol helps to maintain the correct fluidity, meaning it makes the membrane not too liquid and not too solid. And so cells need to take in cholesterol. So cholesterol is produced in the liver. It's going to be moved throughout the body by the bloodstream, and the LDL is going to carry that cholesterol. Now, LDL normally in the body gets out of the bloodstream by binding to the LDL receptor. So the LDL binds to the LDL receptor, and that gets taken into the cell. And so what that means is that you're going to have normal cholesterol levels. So in the bloodstream, you maintain a normal cholesterol level because your cholesterol is able to get into your cells. If you are heterozygous, meaning you have one allele to make the LDL receptor, and your other allele, the little h in this case, is mutated, that little h is the inability to make LDL receptors. And so if you're big H, little h means that you make some LDL receptors, but not as much because you don't have two functional genes. So notice if I look at this diagram, this person would have mild disease. So they would have a slight LDL increase. And that's because they make half as much LDL receptors. So they are able to get some LDL out of the bloodstream because they do have some receptors, but not as much as somebody who is homozygous for the ability to make LDL receptors. on the flip side of this. The flip side of this is if somebody is homozygous for the inability to make the LDL receptors. In this case, it's written little h. What that means is both of their genes are mutated for the LDL receptor. So they're not making any functional LDL receptors. This leads to severe disease. Very high levels of cholesterol. So normal cholesterol, for example, should be, total cholesterol should be about under 200. People who are heterozygous, typically between 200 and 400. People who are homozygous for the inability to make the LDL receptors, their cholesterol can be up in the 1,000. So it's way, way higher than somebody who can make LDL receptors. So for people who have the severe form of the disease, in some cases, cholesterol builds up to such a high amount that you can actually start to get cholesterol deposits, like these little bumps under the skin. If an eye doctor were to look in your eye, they could see cholesterol like crystals forming in your eye. And in the case of the severe disease, patients end up with heart disease and can die in their 20s because their cholesterol level is so high, and that cholesterol starts to clog those arteries and leads to a really early heart attack. And so that would be the severe disease. So again, this would be an example of incomplete dominance. If you are heterozygous, if you have one allele that makes LDL receptors and one allele that doesn't, the phenotype is in between, right? Notice that you make some LDL receptors. So it's not that you have all and it's not that you have none. You have somewhere in between. And that's what leads to the phenotype in between, which is the mild disease. Now, again, I said it's not proper notation to use capital and lowercase when talking about incomplete dominance. So for the purpose of this slide, this again is overly simplified. That is not the correct type of notation that we typically use for incomplete dominance, because again, capital indicates dominance, lowercase indicates recessive. There's not a dominant and a recessive because the heterozygous is in between. So just be aware of that. So what you're going to do is on table 7.1, you're going to look at these traits. So on the left, you have your phenotype. On the right, you have your genotype. And in your lab, there is a description of each of these traits, meaning if we say dark eyes and light eyes, what is different between dark and light? Now, again, you have to realize this is definitely an oversimplification, right? This is very subjective because what I call light eyes might not be exactly the same as what somebody else does. So what I would do is I would start by, for each of these traits, you're going to read through the lab and you're going to figure out which phenotype you have. So for me, I have light eyes. I have mid-digital hair lacking. I have light hair. I have non-red hair. I have a smooth hairline. I have straight hair. I have dimples present. I have one. That still counts. I have freckles present. Long, medium, and short head involves using a caliper and measuring the length of your head and the width of your head. You don't have that at home, so you're just going to cross this entire trait out. You cannot determine which one you have. You don't have access to this tool. Earlobes free or attached. I have free. I have a straight nose. I have a hitchhiker's thumb. PTC taster. PTC is a certain type of chemical, and what we would normally do in class is have you taste this PTC paper and see if you can taste the PTC, which is the chemical that's on the paper. If you could taste it, you would circle PTC taster, and if you could not, you would call yourself a non-taster. Obviously, you don't have access to PTC paper, so you're also additionally going to cross this one out because you can't do that. And so there's a correlation with that PTC tasting and how you perceive foods, if you perceive them as being bitter, for example. And if you're interested, you can read up about that. The next one is going to be toe longer or shorter. So what you're doing is you're comparing your second toe relative to your big toe. So if your big toe is longer or shorter relative to your middle toe. So I'm going to skip that one for now. And you're just going to continue going through all the rest of these traits. So just go through and you're going to fill in all of these traits. Now, we're going to come back in a minute to the normal vision or red-green colorblind, because that is a type of inheritance that we call sex-linked inheritance, right? And so we'll come back to how to interpret that one in a minute. So start by circling all your phenotypes. Now, based on those phenotypes, you're going to circle your genotype. So light eyes, I would be little a, little a. I don't have mid-digital hair, so little b. I have light hair, so little c, little c. I have non-red hair. So what you'll notice is that this is written as big D and a blank. That's because this could be, it's possible that this is big D, or big D, little d. We don't know. So the fact that I don't have red hair, all I can say for sure is that I have one dominant allele. I don't know my other allele. So big D something, I have smooth hairline, which is recessive. I have straight hair. Notice for genotypes for this, right? And also for phenotypes, there's 3. What does that tell you about this type of inheritance if you see 3 genotypes and 3 phenotypes? And the answer is that trait displays incomplete dominance, right? If you're heterozygous, which they have a different notation here, they call it F and F prime. If you have one curly allele and one straight allele, your phenotype is in between, which is going to be wavy. Dimples present, I would be big G something. Freckles present, I would be big H something, and so on and so forth. You would continue down the entire, down through the entire table. Now, class percentages, normally what we would do for this is that we would, after everybody figured out their phenotypes and their genotypes, we would take a survey to figure out what percentage of the class, for example, had dark eyes and what percentage of the class had light eyes. We're not in class. We don't have access to all that data. So for the purpose of this, you're just going to cross the class percentages out. Cross them out. You can't do that. Additionally, there are questions in the lab that ask you about what is the rarest dominant trait, etc. It's asking you about those percentages. You can go ahead and cross those out as well. I believe, and I would have to double check, but I believe that that's questions like 6 and 7. So make sure to double check that number. But it's the questions that ask you about, which of the traits is the rarest dominant trait. You can't answer that because we didn't do our class percentage. So just be aware for those questions that relate to class percentages, cross those out. You're not doing those. So you're going to go through and you're going to fill out your phenotype and your genotype for all of these traits. So example, for each of your traits, circle your genotype. So if you had dark eyes, which is dominant, then you would circle big A something. And if you have light eyes, then you would circle little a, little a. You're going to do this for all 20 of your traits. Then you're going to place your genotype in table 7.2. And I'm going to show you how to do that in a minute. For all dominant traits, for the purpose of this exercise, assume that you are heterozygous. So if you have dark eyes, for the purpose of this experiment, you're going to write that your genotype is big A, little a. Now, do we know that's true? No, right? But for the purpose of this exercise, to show that you could get unique combinations, we are going to assume that you're heterozygous. So again, you might not be, you could be big A, but for this activity, if you have the dominant trait, you're big A, little a. If you have the recessive trait, so if you have light eyes like I do, light eyes would have to be little a. There's no other choice. The only way you see the recessive is if you are homozygous recessive. But if you have the dominant trait, right, then you're going to write that you are heterozygous for this activity. And so you're going to do that for all 20 traits. So let me kind of fill this in for you. These genes over here look a little bit different on yours, but I put this here to simplify it for you so that you could see what the phenotype is. So again, I had circled light eyes. I circled mid-digital hair lacking, light hair, non-red hair, smooth hairline, and straight hair. So based on my table 7.1, then I'm going to write in my genotypes. So if I am light eyes and light eyes are recessive, I'm going to write allele number 1 is little a. Allele number 2 is little a. If I have mid-digital hair lacking, little b, little b. If I have light hair, I'm going to use little c, little c. Now you need to be careful with certain letters. C's, P's, Y, S's. The uppercase and the lowercase look very similar. So like for the C's, for example, if I'm doing this, I would actually, if I'm trying to distinguish between big C and little c, the little c's I write really little. And if I was writing the big C, I would write it really large. And that way I wouldn't get confused about whether it was a little c or a big C. So keep that in mind with letters that look similar. Be very careful to distinguish between them. Non-red hair, again, this is going to be our dominant. So I'm going to assume that I'm heterozygous. Big D, little d. Smooth hairline, so I have the recessive, so little e, little e. And I have the straight hair, so that is going to be the F. I have to go back and look to see if straight or curly was given the prime or not. So maybe the straight was prime. So I'll put F prime, F prime. Double check that one because I'm not looking at that table. But F prime, but I have the 2 straight alleles. So I would do that in table 7.2 for the entire table. Okay, I would fill out what alleles could I give my kids. Now, for this activity, normally in class, you would find a partner and you would record your partner's genotype. So you would have your genotype, you would have your partner's genotype. You don't have your partner available to you. So what you can do is there's a couple options for a partner. You can either use somebody else in your family and use their genotype for the purpose of this activity. Or you could find a picture of somebody that you might be interested potentially in having a kid with, somebody that maybe you find attractive. And you would use their picture to try and figure out as many genotypes as you could. Now, obviously, if you're using a picture, some of those traits, you're not going to be able to look at them, like mid-digital hair lacking. The chances of you finding a picture where you can see that, probably not so good. So for some of this, you might have to kind of make up what your partner's genotype would be. But let's say my partner, for example, had dark hair. I would put big A, little a, because again, whenever they're dominant, assume heterozygous. Let's say that my partner had mid-digital hair lacking, little b, little b. Let's say that my partner had dark hair, big C, little c. My partner has non-red hair, big D, little d, and so on and so forth. So what you're going to do in this activity is this is a create a kid activity, looking at the way that traits are inherited. So what you're going to do is you are going to flip a coin. So let's say I flip a coin and I flip heads. Heads means I give allele number 1. In my case, it doesn't really matter because I know I'm always going to give little a. That's all I have to give. But my partner is big A, little a. So I would flip a coin and I would see what if my partner is going to give the big A or the little a. So let's say my partner flips a coin and they got heads. That means that they're going to give their dominant allele. So the genotype of the child is that I gave the little a and my partner gave the big A. That's the genotype. It's the two genes that they have. Now, when I get to row B, notice that little b, little b, little b, little b. Do I even need to flip a coin in this case? Answer is no, because we know the only choice for offspring, little b, little b. Both parents can only give a little b. So let's say we keep doing this. So little c, little d, and so on and so forth. So you would flip a coin for all of these traits and you would figure out your child's genotype. Then, by looking at their genotype, you're going to determine their phenotype, so the physical characteristic of having that genotype. So if they have big A, little a, they have the dominant trait, which means that their phenotype is dark eyes. They would have mid-digital lacking, so mid-digital hair, lacking. They would have light hair. And little d would be red hair. And again, so on and so forth. Now, couple things to look at. Just based on the first four. Are there any traits that show up in the kids that shows up in this kid that was not found in either parent? And so if I look at eye color, nope, not unique because my partner had dark eyes and I had light eyes. Mid-digital hair lacking, that's not unique, right? Me and my partner both had that trait. Dark hair and light hair, the offspring has light hair. I had light hair, no new trait there. But look when we get to red hair. I was big D, little d, and my partner was big D, little d. So both me and my partner had non-red hair. However, we have a child who is little d, which gives us the phenotype of having red hair. So notice that we now see this unique phenotype that was not present in either parent. That's because both parents carry the red hair gene. You don't see it because it's recessive, but both parents are what we call carriers. They carry that allele, but you just don't see it. And so this is why you could have two parents without red hair and they have a child that has red hair. Or you could have, or you could have two brown-eyed parents and they have a child that has blue eyes. You could have an example. This is actually a dominant trait that you might not be familiar with being dominant. Achondroplasia, which is a type of dwarfism. Achondroplasia, dwarfism is actually a dominant trait. So if you've ever seen the show like Little People, Big World, and you see these two parents that have dwarfism and they have normal height kids, and you might wonder, well, how the heck did that happen? The answer is that those parents that have achondroplasia carry the normal height allele. They have the dominant trait, which is the achondroplasia, the dwarfism, but they carry the normal height gene. And so just by chance, they could both pass on their normal height gene, and you could end up with an offspring of normal height when both parents had achondroplasia. Both parents had the dwarfism. And so that happens because people carry recessive alleles. You don't see them, but that doesn't mean they're not there. They're just not visible. So you're going to do this and you're going to create a kid for all of your traits. You are also, under that table, going to draw to the best of your ability. It's not an art contest, but draw to the best of your ability what that child would look like based on these traits that you're getting. You're going to do that once. You're going to have one kid. Then you're going to repeat it again, and you're going to have a second kid. So there's another column that says child number 2, genotype. So again, flip a coin, record which alleles the child number 2 gets, and then what child number 2's phenotype is. And so you're going to create 2 kids based on your genotype and your partner's genotype. Now, notice that to determine which allele we give, right? And there's a question about this in your lab. If we're looking at which trait we're going to give, notice that the way that we're determining this is by using a coin, right? We're using a coin. I want you to think about what is the significance of using a coin? Why not use, let's say, a 6-sided dice? Why would we use a coin to determine which allele are given to the offspring? And so if you think about it, right, each person, so me, myself, for every gene, I have 2 alleles. There are 2 choices. I could give a dominant, or I could give a recessive, depending on the trait. There's 2 choices, though. So there's a 50% probability that I give one, and there's a 50% probability that I give the other. It's completely random. It happens by chance. Think of flipping a coin. Flipping a coin is completely random. It happens randomly. Sometimes it's going to be heads. Sometimes it's going to be tails. You would expect that about 50% of the time you'd get heads, 50% of the time you get tails. Now, another thinking question along those lines. So we use a coin because the probability of giving a particular allele is a 50-50 chance, right? I could either give allele number 1 or allele number 2. So if I do that though, right, think of a coin, heads and tails. So if I flip a coin just twice, right, if I flip a coin just twice, does it mean that I'm necessarily going to get a 50-50 distribution? Meaning if I flip heads the first time, does that mean I'm going to flip tails so that the probability comes out to be a half? And the answer is no, right? Each event is an independent event. Flipping heads first does not mean I'm going to get tails next. It's completely random. I could flip a coin 5 times in a row and get heads all 5 times, right? And our probability then would not be 50%. It would be 100% of the time I got heads. So when you think about these probabilities, though, you have to think about this is assuming larger sample size. So if I were to flip that coin 1,000 times, so if I flip that coin 1,000 times, because my sample size is large, I would expect that by the time I get to 1,000, that my percentage would be roughly 50%. I would have approximately 500 heads and approximately 500 tails. Think about how this works in human genetics. Think of sex, for example, gender, right? If a couple has a boy, Does that mean they're going to have a girl next just because it needs to be 50-50? And the answer is no. Each child is an independent event. Each sperm fertilizing an egg is a unique event. It doesn't care what happened the time before, right? And so that's why you can have families where they might have, let's say, 4 girls. They have no boys, they keep wanting that boy, and they have 4 girls in a row. Notice that the offspring are not 50% male and 50% female. In this case, they're 100% female. But again, that's because our sample size is small. And so for human genetics, while you expect a particular percentage of offspring being a certain phenotype, what you have to actually realize is that that doesn't always work out the way you expect because your sample size is relatively small. And so just keep that in mind. So again, you're going to do this with you, fill in your genotype, fill in a partner's genotypes. Again, if you don't have a partner, find a picture online, use somebody in your house as your partner. It doesn't matter what your partner, where that information comes from, but you need to put in some data for a partner. And then again, flip a coin to determine a child's genotype, and then based on the genotype, determine the phenotype. And you're going to do this 2 times to create 2 kids. And again, when you're done creating your 2 kids, you're going to draw what those kids look like. So we need to move on and talk about sex-linked genes. So humans perceive light through the activity of specialized cells referred to as rod and cones in the retina. And different sets of cone cells are sensitive to different wavelengths of light, which allows us to perceive different colors. So think of cones, colors. And so there are mutations that affect the ability of subsets of cone cells to register light, will affect the ability of the affected individuals to perceive color. So meaning if they have mutations in particular cone cells, it's going to influence the way that they perceive colors. And so some people are what we call red-green colorblind, which means that they have a difficult time distinguishing between red and green. Some people have blue-green color blindness. They struggle with differentiating between blue and green. And so different types of mutations give us different types of color blindness, meaning that people, it's not that they don't see any color, it's that they have a difficult time perceiving differences between colors. And so if we look at the incidence of color blindness, about 5 to 8% of Caucasian men are born colorblind, and 0.5% of women are born colorblind. And so we're going to talk about why is there such a difference in the incidence of color blindness between males and females, meaning why are males more likely to be colorblind than females? And so on the bottom of the slide, you can see some examples of images that people would look at to try and see if they were colorblind. So if you look at the one on the left, what number do you see in the circle? And you see an 8. If you look at the one on the right, you can see a box, and within it is a star. And so those would help you to determine if color blindness is present. So in order to understand why we have a difference in incidence of color blindness between males and females, we need to talk a little bit about sex chromosomes. And so sex chromosomes are a pair of chromosomes that are used to determine sex or gender. And so if we look at X and Y, so if we look here on the bottom left, what you're seeing is you're seeing an electron micrograph of an X chromosome and a Y chromosome. So during meiosis, X and Y chromosomes, even though they're not technically homologous chromosomes, because remember that homologous chromosomes are matched in size, they're matched in centromere positions, they're matched in the type of genes. X and Y are not technically homologous chromosomes, but they behave as though they are. And what that means is during meiosis, when homologous pairs split, X and Y chromosomes are going to separate during meiosis. So Gametes, or our reproductive cells, are going to carry only one of those two sex chromosomes. Males are XY, so Y has a gene on it called SRY, which is sex determining region. And so the SRY is the gene that determines being male. Females, on the other hand, are XX. So if you think about it, right, males are XY and females are XX. So if you think about who determines the gender of the offspring, is it the males that determine the gender or the females? And so think about that for a minute. Who's responsible for gender of the offspring? And the answer is it's going to be the males, right? Because females are XX. She is always going to give her offspring an X chromosome. So if we look at her chromosomes, she has 44 what are called autosomes. Autosomes are the chromosomes that don't determine sex. And then her sex chromosomes are going to be XX. So when her cell undergoes meiosis, it's going to have 22 autosomes, right? Because there's 22 pairs up here. So her egg is going to have 22 autosomes, and it's always going to have an X chromosome. That's all she can give, right? So the female is always going to donate the X. The male, on the other hand, the male has 44 autosomes, so again, 22 pairs, and his sex chromosomes are XY. So that means that when his cell goes through meiosis, some cells are going to have 22 autosomes and an X, meaning that there's a 50% chance that the sperm has an X, or 22 autosomes plus a Y chromosome. So now what ends up happening is during fertilization, right, when a male ejaculates and the sperm gets into the vagina, the sperm are going to swim up the cervix, up through the uterus, and if they happen to encounter an egg in the fallopian tube, rule for sperm, first one in wins. So if an X sperm happens to fertilize the egg, right, and if the X sperm fertilizes, the offspring is going to be female because the mom gave an X and the dad gave an X. So if the male sperm, so if the Y containing sperm happens to fertilize the egg first, well, then the offspring is going to be male because, again, mom gives X, dad gives Y. And so when you think about gender, males are the ones that are going to determine the gender of the offspring. So when I was having kids, my kid's dad really wanted a boy. And I said, if you don't get a boy, that's on you. That was your responsibility. I don't play a role in this. I always give an X. It's the male who is going to dictate the gender of the offspring. Now, if we talk about our sex chromosomes, our X and our Y, according to recent NIH, National Institute of Health findings, the X chromosome has between 800 to 900 genes. And the Y chromosome, notice how much smaller the Y chromosome is. The Y chromosome contains only about 50 to 60 genes. So the Y has a lot less genes on it than the X. And what you're going to see is when we talk about what we call sex-linked inheritance, most sex-linked genes, meaning most traits that are linked to our X and our Y chromosome, are X-linked, meaning that they're carried on the X chromosome because the X chromosome has a lot more genes. And so we're going to look at a specific example of sex-linked inheritance. So for males and females, again, females are XX, males are XY. So if we're talking about a gene being on the X chromosome, right? If a gene is what we call X-linked, it means that the allele is found on the X. So notice that we have to write this XX, that's the gene. This is the allele. So if we're talking about hemophilia, for example, hemophilia is a disease which leads to a condition where people have problems clotting their blood. So if you're born with hemophilia, you have problems with blood clotting. So if you get a cut, you could potentially bleed to death because you're missing those proper clotting factors that would clot after a cut. Hemophilia is sex-linked and it's recessive. So it's a recessive gene. So for females, if we're talking about hemophilia and we're giving it an H for the hemophilia gene, it's on the X. So an unaffected female could be X big H, X big H. She is homozygous dominant for not having hemophilia. So she clots her blood just fine. On the other hand, we could have an unaffected female, meaning she does not have hemophilia, but she carries the gene for hemophilia. So notice that she is X big H, X little h. So she carries the hemophilia gene, but she herself is not infected. She does not have hemophilia. She is heterozygous and she is a carrier. If we look at females, in order for females to have hemophilia, X little h, X little h, right? So she has to inherit 2 bad copies of the allele in order to be affected. If we look at males, remember, only have 1 X chromosome. So as a result, instead of saying homozygous or heterozygous, we can't say that. They only have 1 X. So we would say that they're hemizygous. Hemi, like think of hemisphere, half. They only have 1 X chromosome. So an affected male that has hemophilia would be X little h, Y. So notice that for a male to have the disease, he only has to inherit 1 bad copy of the disease, 1 bad copy of the gene. Females have to inherit 2 bad copies to have the disease. So this is the same with color blindness. The reason that males are at a higher risk of being colorblind is because a male only has to inherit 1 bad copy to be colorblind, whereas a female has to inherit 2 bad copies. So if we think about males, for example, so let's say we have a male and he has hemophilia. So he's X little h, Y. If he has hemophilia, who did he inherit that disease from? Did he inherit it from his mom or his dad? So think about that for a minute. So if we have a male with hemophilia, who did he inherit the disease from? Mom or dad, or can we not say? Answer is he had to have inherited it from his mom. Because if he's XY, who is the only person that gave him his Y? And that is his dad. So that means the Y came from his dad and the X came from his mom. So if you have a male that's affected, his mom either has hemophilia herself, so she's X little h, and that's why her son has X little h, or she's at least a carrier. She has to be X little h, or X big H, X little h. She's a carrier. She herself doesn't have hemophilia, but she could pass on that X and have an affected son. So when males get it, they inherit it from their mom. Now, let's think of another example. If we have a female who is affected, so if she is X little h, who else in her family definitely has hemophilia? So does mom have to have hemophilia? The answer is no. Her mom does not have to have hemophilia. Her mom could be a carrier, right? Her mom could be X big H, X little h, and by chance could give her daughter the X little h. So this affected female, her mom does not necessarily have hemophilia. Her mom has to at least carry it because that's where she got one of her X's from, but her mom might not have the disease. But if dad gave X little h, that means that dad definitely has hemophilia, right? Because dad would be X little h, Y. And so he gave his daughter his X little h. So males who get a disease inherit it from their mom. Females who have the disease have an affected dad, meaning that their dad had to have the disease in order for the female to also have the disease. Unaffected males, so males that don't have the disease, would be X, big H, and Y, right? We don't write a letter on the Y because the Y doesn't contain that gene. Remember that those chromosomes are not homologous. They don't have the same types of genes. So that gene that's on the X is not present on the Y. So we don't put a letter next to the Y. We only put a letter next to the X. So these are some examples of how sex-linked traits are inherited. And so if we were to look at some examples. So in the example on the left, we have a mom, and this mom is X big N, X big N. And she mates with a male who is infected, who has the disease. Let's say this is color blindness, for example, right? And color blindness is sex-linked recessive as well. So this would be the shaded in. This would be a colorblind, and this is a male because he has a Y. So this is a colorblind male, and he has kids with a normal female who does not carry the colorblind gene. So she's X big N, X big N. So we're going to talk more about these in a minute for Punnett squares. But if we look at the types of gametes that the offspring can form, or that the parents can produce. So dad could either make X little n sperm, right? That's one option. Or dad could have sperm that contain the Y. The eggs, right? The only option for the female and her eggs is X big N. That's one option. She gives one big N. Or again, X big N. So these are the options for the egg. There's two choices. These are the options for the sperm. There are two choices. And then if this sperm is fertilized, so if this sperm is fertilized by this egg, in this box inside the Punnett square, you're going to put both of those alleles, which is why this is X big N, X little n. This XX is female. Does she have the disease? Does she have color blindness? And the answer is no. She's not colorblind because she has the normal allele that came from mom. Same thing with the one beneath it. Mom still gives X big N. Dad gives X little n. This female is not colorblind, right? So from these two parents, the daughters would always be a carrier. 100% of the time. So if they have a daughter, they know that she carries the colorblind gene. I am this example. My dad is colorblind. My mom is not. So because of that, because my dad is colorblind, I'm already female. So I know with 100% certainty that I carry the colorblind gene. I get an X big N from my mom, and I know I have a big N because I'm not colorblind. And I have to have an X little n because my dad has color blindness, right? So I have 100% probability of being a carrier of color blindness. I myself can't be colorblind if the mom is X big N, X big N. But I definitely have to at least carry it. Notice that under this scenario, the males are not affected, right? Because the female, the eggs, always are going to be X big N. So we would have X big N Y or X big N Y. Males are not affected. So with this combination, no possible colorblind children. No possible colorblind children. If we look at the next scenario, mom is a carrier. Whoops, carrier. So mom is a carrier. She is X big N, X little n. Dad has normal vision. So dad has normal vision. So he's X big N, Y. So for the sperm, there are two types of sperm that could form. We could have X big N or we could have Y. And for the eggs, there are two possible outcomes. The egg could contain the X big N or the X little n. So when we put them together, so X big N and X big N, we get a female. She is not colorblind. X big N and Y, we get a normal male. We get X big N, X little n. We get a female who is a carrier. So again, that's why it's colored that like peachish color. And if the female has an egg that is X little n and it gets fertilized by a Y, we have a colorblind male. So for me, I know I'm a carrier. So I know this is me here. And my kid's dad is not colorblind. So he would be this. So now you have to be very careful when looking at genetics problems. And you have to pay very close attention to the wording. So let me show you what I mean by that. So in this scenario, so again, this is me and my ex. And so I'm a carrier of color blindness. My ex does not have color blindness. I have a son. So I have a son. My son's name is Travis. What is the probability that Travis is colorblind? So look at your Punnett square. What is the probability that Travis is colorblind? Now, when people look at this and I ask this question, what is the probability that my son is colorblind? They say 1 out of 4 because they're looking at all 4 possibilities. But that's wrong because of the way I asked it. My son is already born. He is already male. So we do not have to include those probabilities. He can't be those. It's impossible. He's born. He's a boy. So the probability that my son is colorblind is 1/2. 1/2 probability that my son is colorblind. Because he's already born. We already know he's male, so we don't have to include those other possibilities. But now listen to how the question could change, which affects your interpretation. If I were to ask you, so these are the parents, what is the probability that they will have a colorblind son? Notice the difference. Will have. Not already born, not already a male. So if it's asked, what is the probability that the couple will have a colorblind son? We have to include all possibilities. And so now the probability would be 1/4. 1/4 probability that couple will have colorblind son because he's not already born, and so there's an equal probability that it could be female as well. So next one that we can look at is if we have a male who is colorblind. So notice that his box is shaded in. And he mates with a female who is a carrier for colorblindness. She herself does not have colorblindness. Notice that now you can get a colorblind female. And again, this goes back to, I asked the question, if a female is colorblind or the female has hemophilia, which is also sex-linked recessive, Who in her family definitely has the disease? And the answer is Dad. Dad has to have the disease because in order for her to be X little n, she had to get an X little n from each parent. Dad only has one X chromosome to give. So if a daughter has the disease, Dad also has the disease. Mom could have the disease, or mom could be a carrier, but Dad always has the disease. So what you're going to do on your table for sex-linked inheritance is you are going to circle your phenotype. So normal vision. So that means that you can distinguish between the colors. You can Google other examples of looking at images for determining color blindness, or you can use the ones that are in the PowerPoint. But if you could see the 8 in the first example, you have normal vision. I have normal vision, so I would circle this one. If you know that you are red-green colorblind, meaning you have trouble distinguishing between red and green, then you would circle the other phenotype. Now, normal vision. Now I need to figure out my genotype. Again, remember class percentages, we're not doing that. But I need to figure out my genotype. So my genotype is that I have normal vision. So I am X big N something. X little n would be if you were normal vision, normal color vision, or I'm sorry, a colorblind female would be X little n. Or if you are a colorblind male, you're going to write X little n y. You would circle that as your genotype. So I have normal vision though. So I know that I am X big N something. So when I go on table 7.2 to record my genotype, I would record X big N, right? Because I have normal vision. I'm female, so my other allele has to be X. And again, remember that I said that when you're doing this, you always, for the purpose of this activity, assume that you're heterozygous. I am, in fact, heterozygous. I know this, but you might not know this. So, but for the purpose of this, you're going to give yourself a little n. Now, for your partner, even if you do this with somebody at home or a classmate that you talk to, even if you do this with a classmate that is the same gender as you are, don't put X big N, X little n here. That doesn't work, right? That's never going to happen in real life. So no matter what you do, you need to make sure that one person is male and one person is female. So let's say I was doing this experiment and I was working with a partner and she was female and she had normal color vision. Well, I would keep the normal color vision part, but make her male. Or if my female partner was colorblind, right? If my female partner was colorblind, then I would make her X little n Y. And again, I don't put an allele on the Y chromosome. So you would flip your coin. So let's say I got tails first for me and tails again for my partner. My genotype that I would write, X little n Y. What would my phenotype of my child be in this case? And the answer is that this would be a colorblind male. So make sure that you include the gender. This would be a colorblind male. You would flip again for child number 2, record the genotype, and then record the phenotype. And so that is what you're going to do for the first part of this activity, is you're going to look at what your genotype is, what your phenotype is, and then you're going to create 2 kids. So the last part of this video is going to be solving genetics problems. And so I know that you may have covered this in lecture, but I wanted to give you an extra resource and extra tips and ways to succeed in solving genetics problems. So if you already know this, then great. If not, this is extra review for you, or hopefully maybe I explain it in a way that makes sense to you. And so we'll go through some examples and how I would approach solving these genetic problems. Genetics is actually one of my favorite topics. So I put the slide in here. This is just a review of the terms. Again, we already wrote these on earlier slides, so I'm not going to go through all of these definitions. But again, we've talked about these terms in earlier slides. So if we are tracking one trait, we call this a monohybrid cross. So we're looking at one gene. Because it's the same gene, it's written with the same letter and given either a uppercase or a lowercase, depending on if it is dominant or recessive. So let's say we start with the parental generation and we're looking at flower color. And we're crossing what we call a true breeding purple flower. So if you hear that term, true breeding purple, that means that no matter what you do, that purple flower plant is going to give rise to more purple flower offspring. So the genotype would be big P, big P. They're homozygous dominant. The purple flowers is dominant over the white flowers. If you cross that with a white flower, white is recessive, so you know that the genotype has to be little p, little p. So if I cross those two together, the purple flower plant, the only type of gamete that they can produce is big P. The white flower plant, the only possible gamete for that one is little p. So the offspring will always, what's called F1, filial generation, is always going to be big P, little p. They're heterozygous and they give rise to purple flowers. Now, if I take two of these purple colored flowers, so I cross big P, little p with big P, little p. Again, P's look very similar. So for myself, when I write a little p, I put a little tail on it so that I know that that's the little p. So if I cross big P, little p with another plant that is big P, little p, this first plant has two types of gametes. It could either give big P or little p, and this parent can give big P or little p. So when you solve genetic problems, I always start by writing down the phenotypes. So when I read a word problem, I start by writing down the phenotypes first and then determine the genotypes based on the information given. You'll see an example of that in a minute. You need to figure out the different number of gametes that can be produced. So again, if this plant is big P, little p, I know that there's two possible gametes. Either the gamete has the big P or the gamete has the little p. So you need to figure out your gametes. Those go on the outside of a Punnett square. So you draw this box with 4 boxes and the gametes go on the outside. So the big P would go here, little p here, big P here, little p here. And then you're just going to fill in your Punnett square. So big P is going to give rise to some offspring that are big P, big P. Some are going to be big P, little p. And then some are going to be little p, little p. And so notice that if I asked you based on this monohybrid cross, mono meaning one, hybrid meaning they're heterozygous. If I have this monohybrid cross, what I'm going to get is 3/4 of the offspring would be purple. 1/4 of the offspring would be white. And so again, this is just showing you also that when we look at recessive genes, notice we crossed 2 purple flower plants. So both parents were purple flowers. Yet we have offspring that have the recessive phenotype. So we have 2 purples, we cross them together, and we get offspring that have white flowers. What that tells us is that that white flower, it's not gone, right? The white flower allele from the beginning, it's not gone. It's just that we don't see it. It's recessive and it's masked by the dominant purple allele. And so this is again why you could have two parents who have brown eyes and they have a kid that has blue eyes. The parents are both carrying the recessive alleles. You're not seeing it in the parents, but those genes are still there. So this is just kind of a cool idea or a cool slide to kind of illustrate this point. You have this couple and they are mixed race. And so they have a variety of different genes. And so when this couple had babies and they had twins, the two little girls in the dresses are twins. Now notice if you look at their facial structure, you can actually see the resemblance between them. You can see that they look related. But notice how many traits are very different between them. One of them has some of the more dominant traits, so the darker skin, darker hair, darker eyes. And the other twin, just by chance, happened to inherit her parents' recessive alleles. So she has the lighter skin, the lighter hair, the lighter eyes. So what this shows you is this basically shows you that these alleles are retained across generations. Neither of her parents were light-skinned, light-eyed, yet this child is. And that's because some traits are recessive and the parents might carry the recessive alleles, but they themselves do not have the recessive traits because their dominant traits are hiding it. So practice problem number 1. Round seeds are dominant to wrinkled seeds. When a heterozygous round seed plant is crossed with a homozygous wrinkled, determine the probability that the offspring will form wrinkled seeds. So again, when I'm solving these type of problems, I start by writing down phenotypes first. Heterozygous round. So I'm going to write het round, and that's going to be crossed with homozygous wrinkled. All right, so heterozygous round. So I wrote my phenotypes. Now I need my genotypes. Heterozygous round, big R because round is dominant, little r. That's what makes it heterozygous. Homozygous wrinkled is little r, little r. Did I have to tell you homozygous, or could I have just simply said wrinkled? I could have just said wrinkled because the only way that you get that recessive phenotype is to be homozygous recessive. So technically speaking, it did not need to say homozygous wrinkled because if they're wrinkled, you know that they're homozygous recessive. So now it's asking, what is the probability? So probability that the offspring are wrinkled. So this is how I solve these types of problems. So I write out what I'm looking for. All right, so now, parent number 1. The types of gametes could be big R or little r. Those are their gametes. So if I'm making my Punnett square, I have big R, little r. Those are the gametes that that first parent could produce. I'll change my ink. The homozygous wrinkled, the gametes, little r or little r. Notice that's the same. You could just write one little r, and I'll show you that in the Punnett square in a minute. So little r, right, that's the only type of gamete that can form. So I, technically speaking, only have to write it once, because if I fill in my Punnett square, big R, little r, little r, little r. Notice that the probability of being wrinkled, the wrinkled is recessive, is 1/2. If I were to take the time and to write that second little r, right, because little r, big R, little r, little r, 2/4, which is equal to 1/2. It comes out the same. So for me, if I'm crossing with something that's little r, I don't write the second row. It's not necessary, and it's more boxes to count. If the only option is little r, I just write the one little r, and my probability would come out to be 1/2. And so that's what we see. The next question says, an individual with the genotype big A, big B, big C, little c, can produce how many different types of gametes, and what are the possible gametes? So now, there's two ways that you can approach this. If the question had simply asked how many different types of gametes, there's a formula that you can use. And that is that we can do this 2 to the n power, where n is the number of heterozygous alleles. So notice in my practice problem, there's one heterozygous allele, there's another. So my number of gametes possible would be 2 to the second power, because I have two sets of heterozygous alleles. So 2 to the second power is 2 times 2, which is 4 different types of gametes. So if I needed to just simply know the number of different gametes, I can use this formula. The reason I don't have to include this homozygous is that's not going to produce a unique gamete. That's not a unique gamete. The offspring will always have the big A. Now, the second part of this is asking what are the possible gametes. So I'm looking for what are the possible gametes. Now, if you recall back to meiosis, right? The A's, those are a homologous pair. The B's are a homologous pair. And the C's are a homologous pair. And what we're assuming with this type of question is we are assuming independent assortment, meaning we're assuming that those genes are on separate chromosomes, and so they line up independently of one another. Now, for me, the easiest way to determine what are the possible gametes is I like to do what I call a tree diagram. So I start with my A's. With my A, there's only one possibility. It's always going to be a big A. Now, that big A could pair up with a big B, meaning it could line up so that an A has the big B with it, or it could be a little b. Those can line up with big C or little c. And that's true for this one, can pair with big C or little c. So if I write out my possible gametes, I just follow along. So starting at the top and I work my way down. Big A, big B, big C. Big A, big B, little c. Big A, little b, big C. Big A, little b, little c. So notice that I have 4 possible gametes. Those are my 4 unique gametes. Notice that none of those are the same. So when you're doing this, okay, whenever there's 2 options, you're going to branch. If there's not 2 options, then you're not going to branch it. So what I mean by that is, let's do one that's slightly different. So let me write one out that is, if I wanted to know the types of gametes, and we have big A, little a, we'll do big B, and we'll do little c, Okay, so what you're doing for this is, again, if we just need the number, there's only one heterozygous pair. So 2 to the first power, there's 2 possible gametes. Let's look at what they are. So we're going to start, there's 2 choices, big A or little a, right? Those are my 2 choices. Each one is going to pair with a big B, and I'm drawing the line straight across because it's not branching. There aren't two possibilities. It's always going to pair with a big B. This parent's gametes will always have a big B. Same thing, little c, little c. Go straight across, little c. We know it's always going to pair with the little c. The little a could pair with the big B, right? So the big B is the only thing it has, and it's going to pair with a little c. So my gametes are big A, big B, little c. Little a, big B, little c. Again, there are two possible types of gametes. And so this is just a way to figure out what those gametes are. So here is another practice problem. So the practice problem says a cross is made between a purebred green heterozygous smooth plant and a heterozygous yellow smooth plant. What is the probability that the offspring will be yellow and smooth? So again, I'm going to start by writing out phenotypes of my parents. So pure green, het smooth. And so I actually already gave you the genotype for this. So little y, big S, little s. So again, be careful with your y's and your s's. And we're going to cross that, and I'm going to write the other parent in a different color just so we can keep track. We're going to cross that with a heterozygous yellow smooth. So big Y, big S, little s. Now, the other thing I like to do is, because in this problem, I didn't explicitly tell you which one's dominant and which one is recessive. So notice that the green is definitely the recessive. That was the little y. So I like to write myself a note that big Y, any second allele, is yellow. And little y is going to be green. And smooth, so the seeds being smooth, would be big S something. That would be smooth. And little s is going to be wrinkled. So that just helps me keep track of what I'm looking for. So notice that, what is the probability? So I'm looking for, what is the probability that the offspring will be yellow and smooth? So again, that would be big Y, any allele, big S, any allele. So now, notice we're using two different letters. We have Y's and we have S's. Those are two different genes. And again, we are assuming independent assortment. The Y's line up separately from the S's during meiosis. So just like that diagram we showed of the way that they can line up, right? So they can line up differently. So there's two ways that you can solve this type of problem. And I will show you both methods. And you can choose and pick and do whatever method works best for you. So the long Punnett square, okay, the long Punnett square would be that for each of the parents, you need to figure out possible gametes, right? And so for this one, this parent, the possible gametes, little y, big S. Little y, little s. Those are the possible gametes, right? So that would be like if I did my tree diagram. So I'll put it in a tree diagram. So little y can pair with big S, little s. So my gametes are little y, big S, little y, little s. Those are my gametes for the one in red. The one in blue, we have two possible y's. So big Y, little y. That can pair with a big S or a little s. This could pair with a big S or a little s. So this possible gametes would be big Y, big S, big Y, little s, little y, big S, little y, little s. So these are my gametes for one parent. And these are my gametes for the other. So now I make my Punnett square and I put those on the edge. So I'm going to put this parent here. So little y, big S, little y, little s. Those are the two possible gametes. And those compare with big Y, big S, big Y, little s. I probably should have moved that over. I'll move this for now. Big Y, little s. We have little y, big S, and little y, little s. So we're going to fill in our Punnett square. So what we're going to do is, I'm going to put this in purple. So big Y, right? This one gives big, that one gives little. Big S, big S. Big Y, big S, little s. Little y, big S, big S. Little y, big S, little s. Big Y, big S, little s. Big Y, little s, little s. Little y, big S, little s. And then lastly, we have little y, little s, little s. And so again, we're looking for yellow and smooth. So that means Y something, big Y something, big S something. So let's look at ours to see how many of those we have. So here's one. Here's another. Here's another. None of the rest of those meet that requirement because this one is little s, little s. This one up here is little y, little y. So notice that our probability of getting yellow and smooth is going to be 3 out of 8. So my answer for this would be 3/8. Now, for me, this is not my preferred method. And the reason I say that is that one, students tend to get tripped up about what gametes to put on the outside of this Punnett square. You have to make sure that when you do this, that the gametes that are on the outside need to have always one Y and one S. You have to have a Y and an S for each gamete because they're independent chromosomes. So you have to make sure that you have the right gametes on the outside. Then you have to be really careful when you fill in this bigger Punnett square, that you don't miswrite a letter, that you can tell the difference between your big S's and your little s, etc. So my preferred method, the way I like to do this, is a little bit different, and I'll teach you that shortcut. So one of the things that you can do with this type of cross, you have to remember that these chromosomes assort independently. So what you can actually do is cross the Y's separately. So make a Punnett square for just the Y's. So I would have little y is the only option for this one. And this other parent can give big Y or little y. And so my offspring would be big Y, little y, little y, little y. So now I'm just looking at just the probability of being yellow. This is yellow, 1/2. Now I need to cross my S's separately. So I need to cross my S's separately. So big S, little s. And that's crossed with, this one is big S, little s. So again, it's just a very simple Punnett square. Big S, big S, big S, little s, big S, little s, little s, little s. So the probability of being smooth, 1, 2, 3, 3/4. Now, if I want to know the probability of both events happening, meaning they're both yellow and smooth, all I have to do now is use what's in probability called the law of multiplication. And that is that these two probabilities are independent events. So the probability of them happening together, you have to multiply those fractions. So you take 1/2 times 3/4. You multiply the top numbers, 3. Multiply the bottom numbers, 8. Notice I got to 3/8 in a much faster time frame. The same answer works either way. So if you like the big Punnett square, by all means do the big Punnett square. If you would prefer to instead treat them as independent crosses and cross just the Y's and just the S's, that works too. So you just have to figure out which way works for you and go with that method. I personally myself, I don't like the big Punnett square. I would much rather do these smaller Punnett squares and then multiply my probabilities. But it's up to you. So those are both ways that you can solve it, and you can choose which way works best for you. So we'll draw a line. The ones on the left, that's one way. The ones on the right, that's a different way to solve it. You get the same answer either way. So now here's another problem. So an individual with the genotype big A, little a, big B, big C, little c is crossed with an individual with the genotype big A, big B, big C, little c. What is the probability that the offspring will have big A, big B, big C, big C? So this one, you're not going to want to do the big Punnett square. So again, just do these crosses separately. So we'll do A's first. Big A, little a. The other parent can only give a big A, so we only have to draw it once. Big A, little a. And I'm looking for big A, so 1/2 probability of being big A. Then we're going to cross our B's. So big B, so only a big B. And that's crossed with big B, little b. So big B, little b. Probability of being big B, little b, 1/2. And then lastly, we just cross our C's. So big C, little c is crossed with big C, little c. Fill in our Punnett square. So big C, little c, big C, little c, little c, little c. And I'm looking for big C, 1/4. 1/2 times 1/2 times 1/4. So 2 times 2 is 4. 4 times 4 is 16. So we have 1/16. So the probability of getting that type of offspring would be 1/16. And so notice how quickly I can do that if I just treat them as independent crosses and then just multiply my fractions together. So practice problem. PKU is a disease caused by a recessive allele. A man who has PKU marries a woman who does not. So to represent this, I like to use what are called pedigrees. So males are going to be squares, females are going to be circles. So the man has PKU, so we're shading him in. He has the disease. And he marries a female who does not. However, if we look at the woman, whoops, the line got a little crazy there. If we look at the woman, her father had PKU. It doesn't tell us about the mother, but that actually does not matter. So the woman's father has PKU, and the question wants to know, what is the probability that the kid will have a child with PKU? All right, so let's fill in what we know. PKU is recessive. So the man who has PKU, I'm not going to use P's just because they look too much alike. I'm just going to use A's. So if a man has PKU, what is his genotype? If he has it and it's recessive, he is little a, little a. The woman does not have PKU. So I would start out by writing this big A something, right? So the fact that she doesn't have it means that she is big A something. Now, we need to figure out her second allele. We do know that her father has PKU. So her father is going to be little a, little a. What does that tell you is her second allele? That woman, what does her second allele have to be? And the answer is it has to be a little a because her dad had to give her a little a. So without a doubt, she is big A, little a. Now it's just a Punnett square. So little a crossed with big A, little a. So big A, little a, little a, little a. What is the probability that the couple will have a child that has PKU? 1/2. 1/2. So it's a 1/2 probability. Now, PKU, and just in case you're not sure what that is, I'll kind of explain it because it's kind of an interesting disease. But PKU, phenylketonuria, is a disease where people who are born with this is a genetic disease that you're born with, they cannot metabolize phenylalanine. Phenylalanine is an amino acid. And people who are born with this disease, they do not metabolize phenylalanine. And so what happens is they have to be on a low protein diet. Because if they eat protein, they're going to take in phenylalanine and they have no way to break it down. Their enzyme is mutated and it won't break it down. So PKU is one of those diseases that is screened for at birth. If you've ever seen a baby who's had their foot pricked, they're testing for PKU. PKU is a genetic disease, and if it's caught early, if it's screened at birth, and the baby or the child and for the rest of their life is on a low protein diet, then the disease is well managed. The problem is if you don't watch your diet and you have PKU, especially for kids while their brain is still developing, if they happen to eat protein and their phenylalanine levels get too high, that phenylalanine, that excess phenylalanine actually damages the brain and it leads to brain damage in these kids. And so as long as patients know that they have PKU, they have to be on a low protein diet and they have to monitor their phenylalanine levels. It's more important when they're kids. When they get older, the probability for brain damage is less because the brain is not developing. They still have to be careful, but not quite as strict as when they're kids. My ex-husband has 2 brothers who have PKU. And interestingly, just like human genetics, remember, like for males and females, you would expect that if somebody had 2 kids, by probability, half would be male, 1 out of 2 would be male, the other one would be female. But that doesn't always happen. Some families might have 4 girls in a row. Probability of PKU. So interestingly, if you look at my ex's family, so his parents did not have PKU, but they had 3 boys. My ex was the first boy. He did not have PKU, but he had 2 brothers. And they both had it. So the 2 brothers that have it, are little a, little a, little a, little a. Okay. And their parents don't have PKU. So we'll start with big A something. Big A something. Do we know the parent's second allele? The answer is yes. They both have to carry the PKU allele. They both have to have the recessive allele. Neither parent has PKU, but they carry it. And just by chance, these two carriers came together. Neither one in their whole family had ever known that this family carried PKU because nobody else had a child with somebody else who was a carrier. But by chance, these two carriers happened to get together and they had a kid, and they actually had 3 kids, and 2 out of 3 ended up with PKU. That is not what you expect, right? You would expect, so big A, little a, cross with big A, little a. Big A, big A. Big A, little a. Big A, little a. Little a, little a. The probability of having a kid with PKU is 1/4. The probability of having 2 kids, if you have 2 kids, that 2 of them have PKU, that's 1/4 times 1/4. That's a 1/16 chance. Yet, by chance, they ended up with 2 out of 3. So that just goes to show you that because human genetics has a very small sample size, the probabilities don't always work out to what you expect. Yes, you expect only 1/4 to have PKU, but in fact, they ended up with 2/3. So it's just something to keep in mind when you think about human genetics. So the next problem is going to help you with one of the questions in your packet. And that is, so if I write this out. So big apples are dominant over small apples. Red apples are dominant over yellow. You have one tree that produces big yellow. So again, I start with my phenotype that I know. Big yellow, and it's crossed with another tree that produces small red. All right, so when they cross these, they found that half of the trees are big and red, and half are big and yellow. And so this question is asking you, what are the genotypes of the parents? So what I do with this, I wrote out my phenotypes first. Then I go back in and I write out the genotypes that I know for sure. So big, I'm going to start with big A something. I'm not going to fill in the second allele yet. Yellow is recessive. Little r, little r. Small is recessive. So little a, little a. Whoops. The pen's getting a little crazy there. Little a, little a. And then red is dominant. So big R something. So we're going to start with that. Now, the offspring are big. So big A something. Red, big R something. And we have the other plant is big and yellow. So big is big A something. Yellow is little r, little r. All right. So we've written down what we know. Now it's a puzzle. It's figuring out what the other allele has to be. So let's work kind of backwards. So if this parent is little r, do we know what the second allele is for the one that's big and red? Answer, yes. Has to be little r, because that big yellow plant can only give rise to a little r. So we know that that allele has to be big R, little r. The big and red, if we're looking at the A, the big A, do we know the second allele? Yes, right? Because this parent is little a, little a. So we know that's a little a. Big and yellow, do we know what this allele is? Yes, has to be little a, because that plant is small. Now, we need to figure out the 2 missing genotypes for the parents. So we'll start with this one. So if you look at the offspring, you have one offspring that's big R, little r. You have another offspring that's little r, little r. What does the second allele have to be? The answer is it has to be little r, because this child, that offspring, right? Not child, they're not people. This plant is little r, which means they had to get a little r from both parents. Now, if we're trying to figure out this one, right? If we're trying to figure out this one, do we know for sure what this second allele is? Because these are plants and because the number of offspring would give us a decent sample size, we do. We know what the second allele is. And that is, it's not likely to be little a. Because if so, you would expect at some point you would end up with offspring that are little a. But notice you never ever see that. And so because of that, you can assume that this is big A, big A. So the genotypes of the parents are this. Those are their genotypes. So it's basically a big puzzle. Write what you know and then use the information to go back and fill in the parts that you don't know. So there's a question related to this. And I want to give you a few kind of little hints on this. So in a Labrador Retriever, which is a breed of dogs, black fur is dominant, big B, over brown fur. Normal vision is dominant over blindness. 2 black Labradors with normal vision mate and produce 2 puppies, one with black fur that is blind, and another that is brown fur with normal vision. So again, if you're trying to solve this type of problem, I'm going to give you a hint. Start with writing out the genotypes of the parents. So I would write this out, 2 black normal. So black normal is crossed with black and normal. And they have offspring. And they have black and blind. And the other offspring is brown and normal. And so again, just like the question before this, when you're trying to figure this out, for example, if we're looking at black, we're going to write big B something. Normal is dominant, so big N something. And you're going to do that and fill that in for all of this. So question A is asking for the genotypes of the parents. So it's asking you to record what are the genotypes of the parents. You do need to fill in the second allele. If you work through this, you can figure out what the second allele is. Then for B, it says, what are the possible gametes that the parents could produce? Now again, gametes, my hint for gametes is make sure gametes will have only one B, whether it's uppercase or lowercase, and one N, whether it's uppercase or lowercase. But so make sure that your gametes should have one uppercase and one B and one N. So like you could look at the first one, for example, one possible gamete, big B, big N. That's a gamete. It only has one B and one N. So make sure when you're talking about gametes, they only have one of each letter. If you're talking about the genotypes, what are the 2 possible genotypes of the pup with black fur? That's this. What are the genotypes for this black pup? What is the genotype for the brown fur? The genotype is going to have 2 B's and 2 N's. And what you're going to find is that there's 2 possible genotypes that they could be. You have to work through this on your own. Now, E is asking you to do a cross between the 2 parents, the black normal and the black normal. So once you get your genotypes for the parents, you're going to cross those parents and record what is the probability that the puppies would be black with normal vision. F is if additional puppies were produced, what is the probability that they would be brown and normal? So again, that's using your Punnett squares to figure out the probabilities. And so that's how you're going to solve this type of problem. And so use the examples from earlier to help you kind of work through this. But I wanted to at least kind of help set you up and ask you and show you what they were looking for the different parts. Because again, sometimes students get confused with the genotypes of the parents, the gametes, etc. So the genotype, so this is A up here, this row. Okay. And so that's what you're doing for this question. For question number 10, why did you toss a coin to determine which allele was passed down? So again, we talked about that earlier, so go back and listen to the video. Why use a coin versus, let's say, a 6-sided dice? B, does the child number 1 look like you or your partner? So look at the traits of the child and compare that with your traits and compare that with the traits of your partner. C, does child 2 look like you or your partner? Same thing. D says, are there any phenotypes that either child has that neither parent, that neither you nor your partner has? The child might have light eyes and both parents have dark eyes. If yes, which one? So in the example that I did in class, my partner and I had non-red hair, but we had a child that had red hair. So I would write that here, red hair. And then E, how can a child have a phenotype that is absent from either parent? And so I've talked about this numerous times, so hopefully you can fill that in now, right? So again, that's like our 2 brown-eyed parents having a child that has blue eyes, or 2 people that do not have PKU and they have a child that has PKU. How does that happen? That's what that question is asking. And so you can read through this. This is basically your study guide to help you study for quiz 7. And you're going to read the next lab.